Hello everyone. Today we're going to be learning about stem cells. This is our last lesson in the cells topic. So once we've done this, we will be revising for the topic test. But today we're going to spend our double lesson on stem cells. So date and title, please, and pause the video for a couple of minutes to get your keywords down, and you'll do now task finished. Sorry. Let's move me out of your way. Okay, I'm going to move on. So let's talk about the do now tasks. Differences between mitosis and meiosis. Well, hopefully you remembered from yesterday that mitosis is cell division for the purpose of growth and repair. It only produces 2 daughter cells, whereas meiosis produces 4. And the purpose of meiosis is to produce gametes or sex cells, such as sperm and eggs. Also in mitosis, the daughter cells are genetically identical to the parent cell, whereas in meiosis, they are genetically different from the parent cell and actually genetically different from each other as well. You might also have said that in mitosis, the cells just divide once, whereas in meiosis, there are 2 stages of cell division. So one cell divides into 2, and then those 2 cells divide again to give us a total of 4 for meiosis. What happens when you have faulty cells? Well, if faulty cells are dividing, then that can cause uncontrolled cell division, and that leads to cancerous tumours. Okay, let's move on to stem cells then. So, cell differentiation. What do we mean by that, first of all? Well, we've learned about specialised cells, different types of animal and plant cells, but before a cell becomes differentiated or specialised, it's known as a stem cell. So cell differentiation is the process that a stem cell goes through in order to become a specialised cell. So if we take humans, for example, our bodies are full of specialised cells, muscle cells, nerve cells, bone cells, etc., etc. But in certain parts of our body, we've got undifferentiated cells known as stem cells, which are there sort of waiting to make more specialised cells. So a good definition to write down is a stem cell is an undifferentiated cell that has not yet become a specialised cell. So you could pause the video here to get that written down. Other features that are worth noting: stem cells can replicate many times. So stem cells can replicate to make more stem cells if that's what your body needs, or a stem cell can differentiate and become a muscle cell, for example, and then it will divide by mitosis to form new muscle cells. So stem cells have the potential to become other types of cell. So they're really important cells. Video clip to show you now. While you're watching this, think about where is it that they say stem cells come from, and what medical conditions can stem cells potentially be used to treat.
It's nothing more and nothing less than a small bag of baby's blood. Dr. Kurtzberg is one of many doctors who believe that the tiny quantity of blood taken from the umbilical cord and placenta of a newborn baby has the power to build an entire new blood system. They call it cord blood, and the reason she's so sure cord blood can work is because it's full of blood stem cells, the key to life. These are stem cells dividing to make new blood cells. What makes them so special is that they have one truly unique capacity. They can create any type of blood cell that we need. The white blood cells that fight infection, and the red blood cells that carry oxygen. Blood stem cells are very rare, yet they are the factory of our entire blood system.
The normal blood formation is an incredible process. It's making tens of millions of new blood cells every hour. All of those cells are coming from a fairly small number of rare stem cells, which are going to continue this process over weeks, months, years, and decades of an individual's life.
But when the blood system becomes seriously diseased, the only thing to do is rebuild a new blood system from scratch. This means replacing the patient's faulty stem cells with healthy ones from someone else. For years, science knew of only one source of these special stem cells: the bone marrow, a jelly-like substance inside our pelvis and other large bones. It's the home of stem cells. Until recently, the only treatment for critically ill children was a bone marrow transplant.
Okay.
Did you know that...
So where do stem cells come from? Well, in the video they talked about stem cells being taken from the umbilical cord of a baby. That's the cord that came out of your tummy button when you were in the womb inside your mum and joined you to your mum's body. And also from bone marrow. So in adults, and when we say adults in stem cells, we're actually talking about a fully grown human. So that includes babies. So not an embryo, not a fetus in the womb, but a fully formed individual. So those stem cells come from places like large bones that contain bone marrow. So 2 types of stem cells were mentioned: adult stem cells from the bone marrow and embryonic stem cells. They mentioned the stem cells from the umbilical cord. Those would also be classed as embryonic stem cells. So embryonic stem cells usually come from developing embryos. So embryos are fertilized eggs. So when a woman first gets pregnant, and there's that tiny little bundle of cells growing in her womb, as that forms something resembling a baby, we call it an embryo. People who struggle to get pregnant can have various types of fertility treatments to help them, and one of those is called IVF. IVF is, if you've heard of test tube babies, that's basically what IVF is. IVF is where you take sperm from a man, eggs from a woman, and in a science lab, they actually mix the sperm and the eggs together. So the sperm fertilizes the egg. They start to grow that embryo, and then any healthy embryos could be put back inside a woman in the hope that she will become pregnant. So in this treatment, usually lots of sperm and lots of eggs are taken, lots of embryos are made, and then the most healthy ones are selected to be put back into the mother. And that means there are leftovers. And nothing sort of happens to them. They're generally put in a freezer, and then eventually they might be destroyed. But scientists can use them, only with permission of the parents, but scientists can use leftover embryos in order to extract stem cells. Next video.
Our bodies are the ultimate factory. Every cell has its specific job to do, and is shaped to do that job perfectly. A muscle cell is a muscle cell, and only a muscle cell. A nerve cell will only ever be a nerve cell. And a red blood cell will only ever be a red blood cell. The fate of each cell is determined during the embryo stage, and then cannot be changed. However, cutting-edge research made a major discovery for modern science. There are these wonderful cells that exist called stem cells. These cells have the ability to become any type of cell because they are not specialized. Scientists were hugely excited by their discovery because they can be used to repair damaged organs, bones, or cartilage. They could also be used to help treat diseases, such as by making new brain cells to treat people with Parkinson's disease, or they could be used to repair damaged immune systems and even reverse paralysis or regrow lost limbs. The discovery of stem cells is huge for modern medicine and could have a massive impact. There are two types of stem cells in humans: embryonic stem cells and adult stem cells. Embryonic stem cells are unspecialized cells that can develop into any type of cell. Adult stem cells are also unspecialized cells. Whilst they can develop into many types of cells, unlike embryonic stem cells, they cannot develop into every type of cell. Adult stem cells come from tissues such as bone marrow. As in the name, embryonic stem cells can be removed from unused embryos, such as those left over from fertility treatments, for example. Whilst the discovery of stem cells offers huge potential for modern medicine, there are many social and ethical considerations that need to be taken into account. A lot of research was, and still is needed, to find out exactly how they work and how we can control them to do what we want, and not just grow uncontrollably. Stem cells are already being used in treatments. Currently, in 2016, there is a trial study underway to use stem cells to reboot the immune system of multiple sclerosis sufferers to enable them to regain some mobility. Using stem cells to treat diseases and conditions of the blood and immune system are currently the most widely used stem cell treatment. Over 26,000 patients are treated with blood stem cells in Europe annually. A new stem cell treatment has recently been approved for repairing damage to the cornea. Stem cell treatments are also being used in rhino conservation efforts, as well as other endangered species. One big social and ethical debate around stem cells is therapeutic cloning. This is the concept of cloning a patient, growing an embryo from the clone cells, and then extracting stem cells from the embryo to grow a specific organ needed by the patient. The patient's body would not reject this organ, as it is made from the patient's own cells. The ethical debate focuses on defining when does life start. Is the embryo alive? People also don't like the idea of us playing God. We will look at the process of cloning in another video. Unspecialized stem cells also exist in plants and can develop into the cells of flowers, roots, or leaves. Unlike in animal cells, some plant cells do remain unspecialized and can then develop into any type of plant cell. The unspecialized plant cells group together into structures called meristems. Whereas animals stop growing in size when they are adults, plants do not. This is because meristems divide to produce cells, thus enabling plants to continue to grow both in height and width throughout their lives. They also produce cells that develop into flowers and leaves. So from this video, you should know that stem cells are present in embryonic and adult animal cells, and in meristems in plants. Stem cells are unspecialized and can develop into a range of different cell types for development, growth, and repair. In animals, embryonic stem cells can turn into any cell type, whereas adult stem cells, which are found in bone marrow, can only turn into some cells.
Okay, so there was a lot of information in that video, and I'm just going to recap some of the main points. In fact, let's just find that bit again.
Any cell type, whereas adult stem cells, which are found in bone marrow, can only...
rats. Okay, so here's some of the key things we need to take from that. So stem cells, embryonic and adult cells are found in animals, but in plants we have stem cells in what's known as meristems, which are parts of the plants that can actually grow new roots, leaves, branches, etc. If we cut an arm, we can't just grow one back, but if you cut a leaf off a plant, it can grow a new one because it has these areas called meristems. Stem cells are unspecialized, so they can develop into different cells. Now, embryonic stem cells can turn into any type of cells, but we can only get them from embryos. So unused embryos from fertility treatments would be an example of where we could get those from. But some people really object to the use of embryos. They feel that even if an embryo is not going to be used in fertility treatment, it's still a potential life, and it was still made by two people, and it's not something we should be destroying in order to obtain stem cells. Other people would argue that actually the medical benefits to people who can have treatments using stem cells far outweighs the problems of destroying an embryo. Adult stem cells, which was the next bullet point on the screen, adult stem cells can generally turn into 1, 2, or maybe 3 types of cell. So they're useful, but they're not as useful as the embryonic stem cells. Of course, they're much easier to get. We can extract them from people. There was also a mention of something called therapeutic cloning. Just to be clear, when therapeutic cloning is carried out, and actually it's not legal yet, but that would involve cloning a cell from a human in order to extract stem cells, not cloning the whole human. So they would just grow a very small, like an embryo, from somebody's own cells in order to extract the stem cells. Okay, they also mention lots of medical treatments, and I just want to point out that whilst there are some medical treatments available using stem cells, most of the stem cell work that's being done by scientists at the moment is still in the research phases. So there aren't that many medical treatments that have been approved yet, but stem cells have the potential to treat lots and lots of diseases. We could potentially grow new organs for people using stem cells. Okay, so just if your teacher can pause the video here, just to let you think through these questions. If you've already written down the information, you don't need to write it again. Just go over your notes and make sure you understand it. If you haven't got this written down yet, answer the questions in your book. So to go through the answers now, stem cells are undifferentiated cells. 2 types would be adult and embryonic. Adult stem cells are found in bone marrow, and embryonic stem cells are found in embryos, such as leftover embryos after IVF treatment. And if you can pause the video again and just copy these sentences into your book, filling in the gaps with the keywords at the bottom, and I'll show you the answers in a moment. Here's the answers. So again, the video can be paused here just to let you check your answers, mark your work, make any corrections. And moving on. Okay, so moving on to your next task. You should have some printed information, key points about adult and embryonic stem cells. So just in case you haven't got them, they are on the next slides. I'll go through them in a moment. What you need to do is look at the information, and for embryonic stem cells and then for adult stem cells, categorize the points into benefits of using those stem cells and problems or risks or disadvantages of using those stem cells. So I will just go through the points. So extraction of embryonic stem cells is painless. There are lots available, but although there are lots of embryos available in laboratories, embryos can't be asked, and people have ideas about embryo rights, and they feel that actually the embryo has the right to be preserved. We do harm or kill the embryo when we take stem cells. There's no getting away from that. Stem cell treatments using embryos are still relatively new and untested. So even if you are offered one, there's every chance it won't work. They could potentially treat a wide variety of diseases and medical problems, but we don't yet know some of the long-term effects because it's very much in the early research stages. So there are risks associated with having those sorts of treatments. Some people would argue that using them is better than wasting them, and there's a fear that embryos could kind of be something that people go into business selling, and some people feel that's really not right. Your adult stem cell key points. There aren't really any ethical issues in using adult stem cells because you ask the person's permission before you remove them. You would never just take stem cells from somebody by force. We've done lots of treatments with adult stem cells already, so we know which treatments work. They only adult stem cells only turn into a few types of cells or tissues, so there's only a few diseases or problems we can treat using them. Extraction of them is uncomfortable. It's like a minor operation, so some people wouldn't volunteer to donate. It's fairly safe, but there are some risks. With any medical procedure, there's a risk of infection. Finding donors can be difficult. Sometimes there's problems with rejection, but you do generally make a quick recovery from stem cell treatments that use adult stem cells. So if you don't have those printed out, then the teacher can just rewind and sort of pause the video with those on the screen for you. That is your task. So if the teacher can pause the video here, I would think this will probably take you 10 to 15 minutes to do this. Okay, I'm going to move us on now. So hopefully you have managed to work out the benefits and risks or problems associated with embryonic and adult stem cells. Your next task is to produce a fact sheet detailing the structure, location, and potential of using stem cells. So what do we mean by a stem cell? Where are they found? What is their potential for medical use? In other words, what sort of diseases or medical problems could they treat? What you think ethical issues are surrounding the use of stem cells. The mind map that's on the screen gives you a sort of criteria as to the sorts of things that you can include. And actually, if you'd like to lay your fact sheet out as a mind map, that would be absolutely fine. After this, you're going to do a written piece of literacy task. So this fact sheet will really help you with that. It can be short and punchy. It doesn't need to be full sentences. Bullet points is fine. This is really just to organise the knowledge that you've gained today on stem cells so far. So again, I would think this will probably take 10 to 15 minutes to do. So the video can be paused while you get on with that. Okay, we're going to have a look at an exam question now. So remember, with exam questions, use your TICS formula to help you structure your thinking. So when you read an exam question, think about the topic. Well, it's stem cells and cell division. Think just in your head, think through the important information you've learned about this topic and your core knowledge. It's worth thinking about the keywords you might want to use answering this sort of question. And then the next stage is sort of how to start your answer. So you can just jot your answers down to this. So stem cells divide by mitosis and new specialized cells develop. So we've got a diagram of a stem cell that is becoming lots of more stem cells by mitosis and then is turning into types of specialized cells. Suggest one way doctors can use stem cells to treat patients. So just jot down your answer to that. Sorry, I said your next task would be the literacy task. I'd forgotten about this exam question. We'll do this first and then we will move on to the literacy task. Next question. Stem cells from embryos can be used in medical research. Explain why some people object to this. This is a 2-mark question, which would suggest that you might want to make a couple of points. I'm going to move on. The video can be paused if you haven't finished. Another question. I'll read this out because it's very small. The drawing below shows human embryonic stem cells. What features of stem cells could make them useful in treating many different medical conditions? I'll repeat that. The drawing below shows human embryonic stem cells. What features of stem cells could make them useful in treating many different medical conditions? I'm going to read the next one now, so pause if you've not finished. Next question. A laboratory in Japan has recently developed stem cells from human adult skin cells. Suggest why using human adult stem cells might be preferred to using human embryonic stem cells. So I'll repeat that. A laboratory in Japan has recently developed stem cells from human adult stem cells. From human adult skin cells. Suggest why using human adult stem cells might be preferred to using human embryonic stem cells. And the next question. Plants also have stem cells. Which 2 numbered parts, 1 to 5, of the plant shown above contain stem cells? So number 1 is the root. Is the tip, the growth tip of the plant. Number 2 is the leaf. Number 3 is the leaf stem. Number 2 was the tip of the leaf. Number 4 is the leaf. And number 5 is the root. So which 2 areas? Think about which parts of the plant are going to be going to be growing the most, and perhaps developing new bits of plant rather than just getting bigger. Okay, let's look at the answers now. So first question, replace damaged tissue or cure diseases. Next question, it involves destroying embryos or the potential loss of life. They are undifferentiated cells, for 1 mark, that can specialise into any type of cell, 2 marks. It's the any type bit that gets you that 2nd mark. Next question, destruction of embryos. And lastly, 1 and 5. So that was the tip of the plant and the root of the plant. But if you've just written numbers for your answers, that's absolutely fine. Okay. This is the last task I'm setting you for this afternoon. I know this is a long double lesson. If you finish before the end of the lesson, then please start doing some revision work on this topic. So perhaps grab your book, and if you're not working from your book today, and look back over the various topics we've done. You could start to make some revision notes in the back of your book, perhaps or on paper. But first, the literacy task, and you may well need the rest of the lesson to do this. So the more I read about stem cells, the more I chop and change my mind about the uses of embryonic stem cells. That's perfectly genuine. Sometimes I think it's a really good idea, and other times I really struggle with it. I find some of the ethical issues quite difficult. So, referring in detail to the knowledge you have gained today, write me a persuasive letter with the aim of persuading me either that embryonic stem cells should or shouldn't be used in medical research and for medical treatments. So you can pick whether you're going to write a persuasive letter that is for the use of stem cells or against the use of stem cells, but I want you to use the information you've got today to persuade me one way or the other. Now, you're going to need to make reference to both arguments, but I want you to explain why you think one argument outweighs the other. Now, if like me, you're a bit undecided, then what I'm going to ask you to do is just choose a viewpoint and practice arguing for that viewpoint over the other one. Okay, but it's totally up to you which viewpoint you take. So this is the end of the video for today. This is your last task for today. If you finish early, then on to some revision, please. I hope to see you soon.